Right, let's get the rest of this done. So, yeah, you can see on the diagram, I always put on my N and 2N type things. So yeah, I've got 2N, oops, there. That's a bad start. Pick a pen. Yeah, so zygote must be 2N. Fertilization, zygotes fuse, so it must be 2N. And therefore, what it's telling me here that it's N, so I know that meiosis happens at R. And therefore, yeah, the gametes must still be N. And gametes have to be N because they're going to fuse as in haploid. And on B, yeah, I can see this dividing arrow here. So yeah, so the gametes must be N because I get my fertilization and 2N. So yeah, you can work out it's diploid haploid on 1.1 and which row in the shows where meiosis occurs in each life cycle. Well, I've kind of like done it already. Yeah, again, by annotating the diagram, I can work out its R and S. It's nice and easy. 6.3 is tricky. Again, I've drawn a little diagram to help me. But yeah, this is in the first stage of meiosis. I'm going to have 4 chromosomes going one way and only 2 going the other. And then they're going to split later on to give me 4-4-2-2. Sounds like football. 6.4 is straightforward, but look how I've set it out. Yeah, numbers of pregnancies equals 7,000, and then probability of downs is that. Again, it's setting out is crucial. There's not a lot to do here. RER, yeah, X. I know it says, also, a cell membrane is a structure, so it is X. I've not done 7.3 because I can't measure on this iPad, but you should be measuring the scale bar in millimeters, multiplied by 1,000, and then divide by 10. Yeah, nice and easy. And why do they need lots of these for Z? Well, they've got loads of RER and Golgi, so they're obviously secreting proteins. So, yeah, Z is a mitochondrion, sorry, site of aerobic respiration, make lots of ATP, and then it's what it can be used for, either vesicle transport. You could say, the actual direct role of ATP is to charge tRNAs with the suitable amino acids. But ultimately, that energy goes into a peptide bond. So I'd always say that eventually, anyway. So that was straightforward, I think. 8.1 is just knowledge, left ventricle. Yeah, stronger contractions can produce more pressure for a thicker wall. On this one here, yeah, the pressure is decreasing in the ventricle. Therefore, blood must be leaving. So if blood's leaving, it's going out of the semilunar valve. And to make sure it doesn't go back into the atrium, the AV valve needs to be closed, the atrioventricular valve. Explain the shape of the oxygen dissociation curves. Yeah, so why is it shaped like an S going upwards? So I've done the S bit first. So I can just point out that, that when hemoglobin binds an oxygen, it changes the tertiary structure of hemoglobin, making it easier for the next oxygen to bind. That's why we get this S shape. And then it's just pointing out that high oxygen concentrations or high partial pressures, I should say partial pressure because it says so in the question, hemoglobin becomes fully saturated, or it's highly likely to become fully saturated. And at lower partial pressures, it dissociates from the oxygen and releases oxygen. So again, straightforward. 9.2, I think I was one out compared to the mortar scheme. But yeah, I highlighted the mouse and the cat. It's an awfully printed graph, apologies. But yeah, take one from the other and get 5%. I think it might be 4%. I think the cat might have been 50, not 51, but I think by the time I highlighted the line, I'd got myself confused. But the principle is there, and it wouldn't be that badly photocopied in an exam. Explain why the curve is to the right-hand side for a. Explain why the curve for mouse is to the right of the curve for elephant. Yeah, so as soon as it goes on to the right hand side, it's the same as the Bohr shift. So I can say that the mouse, it must be the mouse's hemoglobin, not the mouse has lower affinity. I have to say hemoglobin has a lower affinity for oxygen. That means it releases more oxygen to allow faster aerobic respiration. And then what's the benefit? Well, mouse is smaller. It's got a higher surface area to volume ratio. It will lose more heat. And therefore, by doing more respiration, it's going to release more heat to stay warm. I've done my green line. Yeah, so it's just if it's at high altitude, it needs to it has to be to the left so it can become fully saturated at lower partial pressures that will be present there. Then this organic farming, they're not using insecticides, herbicides, etc. Species richness is just the number of species. So I've got one here, one there, one there, and one there. It's a conventional farm. That's that one. So it's 4. That's good. I'll give myself a tick. And then the slog of working out an index of diversity or diversity index. So remember this, the total of the numbers given, that's big N. So I've got my 117, and I have to knock off 1. So it's multiplied by 116. And then I have to multiply. I always write the -1 next to all of them. I have to multiply them all together. So that's N multiplied by my N minus 1. So I've written in my N minus 1. And then the sum of is there. So that comes to 2278. And therefore, oh yeah, they give me the conventional form to 2dp. So I'm giving that one to 2dp. And this is, anything to do with like why they got rid, like they've taken down hedgerows and things. So yeah, but it's told me why the organic farm is better than the conventional farm. So there's no information about hedgerows, but they have told me that they've used herbicides. So that's where I'm going, because herbicides are weed killers. So there's a higher diversity of plants. So there's more different species. That's what diversity is, because they've not been using herbicides. And this is the crucial bit. Yeah, there's more different niches, places to live and feed. So I get a greater diversity of butterflies. There's lots of ways of answering that question. F, yeah, leaf cells and white blood cells have a nucleus, and leaf cells and prokaryotes have a cell wall. And what's in space? D, yeah, all cells have got cytoplasm and ribosomes, but it says organelle, so it has to be ribosome. And G could be permanent vacuole, but I put chloroplast. I'm still correct. Oh, we're romping through this. Again, this is my working out. So yes, I put my kingdom, phylum, class, order, family, genus, species down. So then this becomes just straightforward to do those 2 questions. And then it's an evolve question, 4 marks. So I always teach vibe, which is variation due to mutation. Good. I stands for intra-specific competition. So these mayflies are competing with the rest of their species. Well, those that are green will have better camouflage, and therefore they'll have less predation. But I have to talk about an allele. Those with an allele for green colour. And then these green mayflies survive to breed, critical I say that, and pass on their green-causing allele to their offspring. I need all those 3 words: breed, allele, offspring. And then evolution is a change in allele frequency. So that the frequency of the green-causing allele increases in the population. Population is vital as well, and you do need to say increases, because it would increase, because they are changing. The population is becoming increasingly green. Maybe there's been a bit of climate change. It's got a wetter environment, more grass is growing, and that's driven the evolution of the mayfly. 13.1, it can't be endoexocytosis, because that's vesicles fusing. So is phagocytosis, so it has to be facilitated. Yeah, and cholesterol does control the fluidity of the membrane. So just facts. Why does it form a bilayer? Again, hopefully you said about hydrophobic, hydrophilic. I have as well, but yeah, you have to get to the fact that there's water on both sides. Therefore, the charged heads always face outwards. And why can't you see this structure? Well, it's because the TM uses electrons that don't provide the resolution. their wavelength, as in the electrons. I'll put that down as well. There, as in electrons. People always make it sound like a microscope has a wavelength. Yeah, so electrons' wavelength is too long. You're usually saying that electrons have a short wavelength, but it's not short enough. So I could say it like that as well, not short enough. Then we've got our plant. It's been fed radioactive carbon dioxide. It's transparent box, so it's getting light. Can do photosynthesis. And then it's got a bud at the top, brilliant. It's got roots at the bottom. Then the plants were collecting bright light for an hour, right? So it's done photosynthesis. And then the reactivity was measured at 4 positions. Yeah, place an X on the ringed plant. Well, the ringed plant is the one that's going to have no transport. So yeah, my leaf is going to be radioactive, but here there's none, so it must be between the leaf and there. So I put it anywhere, kind of like between in this area. I can't put it there because that would cut off sugar to the rest of the plant, so that's not allowed. So it needs to be in the main stem between those two areas. And explain how radioactive carbon dioxide from CO2 reaches the bud at the end. So again, I'm saying, yeah, that it carries out photosynthesis, uses the carbon dioxide. I'm going to convert it into sucrose. That's going to be like carbon 14 containing. I put too much information here, but you never know. So yeah, sucrose is actively transported into phloem. Don't really need sieve tubes, or don't need sieve tubes. This lowers the water potential, causing water to enter by osmosis. Pressure builds. And I think the marking point was translocation, but I've given a reason how translocation works. It's never lost. And then why is it more radioactivity in the bud? Well, the bud is storing the radioactive sucrose or using it for growth, whereas in the stem it's just transporting it, it's just accumulating. Again, you never know, so I've carried on going. If it was using some sugar to fuel respiration, because the phloem carries out respiration, then it'd be losing the radioactivity as carbon dioxide would be diffusing away. And explain the results for the ring plant. Yeah, it's just knowing that ringing removes its phloem tissue, and that's why it's been unable to get down to the roots. That's another nice easy one. Oops, we're almost there, flying through. Then we've got our question again. I've just kind of I underlined numbers. They weren't actually needed because this is just saying like it's like background. So this 0.5 is that 0.5. It's describing this first experiment. That's my 10 and that's my 10, right? So it's just I'm just confirming it. I know the stuff. How do active site of enzymes work? So it's kind of wrote it in a bit of a backwards way, but yeah, it uses the induced fit model. It molds around the substrate to lower the activation energy. When it forms ES complexes, it means it's easier to break bonds in the substrate. What other things would they could have controlled? You could put temperature down. I put pH, or anything about the potato, age, yeah, yeah, probably not size of potato, but you can say like age or the I don't want to say species of potato. I want to say like the breed of the potato. Anyway, stick to pH or temperature, and then complete the table. This is actually tricky because it's like, what would you actually do here? I've got a couple of options, but you would make the fastest equal 1. If I make the fastest equal 1, because it's arbitrary units, it's not saying, it doesn't say, like seconds to the -1. So it can't be just do 1/6. That's what I was tempted to do. But yeah, the arbitrary units means it's like, well, what would I actually do if I wanted arbitrary units for it to make sense? So yeah, I'm basically dividing 6. I'm going to say like, yeah, the shortest time is 6, and therefore I'm going to divide by 6. 6 divided by 6 is 1. 6 divided by 6 is 1. 6 divided by 18 is 0.3 recurring. 6 divided by 10 is 0.6. and then 6 divided by 7 is 0.86. Why have I done 2 dp? I was worried about this one here. I mean, it did round to 0.9, but it'd be a bit of a leap in a graph. So yeah, 2 dp is more sensible in this one because it means I get fine discrimination between all these values. And that is it. I think I'm getting faster at this. Super. Good. I'm going to stop.